Welcome to Chapter 3. In this lecture, we will address 11 objectives and questions. We will start by simply defining a cell, as well as looking at the three major regions of a generalized cell, and also outlining their functions. We will look at how substances or molecules enter and exit cells in the human body, and then we will finish up by looking at the organelles of a cell, or the little organs that allow cells to function. Going back to the structural or hierarchy organization of the human body, we saw that the chemical level was the smallest level, but once we put atoms, molecules, and organelles together, we created the cellular level. We know that a cell, or the cell, is really the smallest unit of life, and that all organisms are made up of one or more cells. Overall, a general cell will have three basic parts. Those are listed on the screen. We have the plasma membrane, the cytoplasm, and the nucleus. The plasma membrane will be like the fence or the wall that's responsible for keeping things out and holding things in. The cytoplasm is really everything in between the plasma membrane and the nucleus. And then lastly, the nucleus is really the command or the control center of a cell. Pictured on this slide here is a general cell. Looking at the image, you are able to identify the three main components of a generalized cell in the human body. You are able to point out the plasma membrane, which again is that flexible outer boundary, which holds things in, keeps things out. In between the plasma membrane and the command center, also known as the nucleus, we find the cytoplasm. So there's a lot going on in the cytoplasm that we will cover here in just a bit. For the next portion of this lecture, we will talk about the plasma membrane. We will address its structure as well as outline many of its functions. As previously mentioned, the plasma membrane is that flexible outer boundary of a generalized cell in the human body. It's responsible for separating stuff outside of the cell, known as extracellular fluid or extracellular material, separating that from whatever is inside of the cell, known as intracellular fluid. Outside of the cell, you might find interstitial fluid, which is tissue fluid. Blood plasma is surrounding red blood cells. Cerebrospinal fluid in the nervous system, so on and so forth. We will see that along this boundary or membrane, there are many doors and windows that will allow for certain substances and molecules to enter and exit the cell. We refer to the plasma membrane as a phospholipid bilayer, and that's because the majority of that membrane or of that boundary is primarily phospholipids. These phospholipids sort of resemble a jellyfish or a lollipop, with the head of the jellyfish or the main part of the lollipop being the hydrophilic head, and then the stick or the tentacles being the hydrophobic tails. Hydrophilic means water-loving, so the outside of that membrane is okay being in direct contact with water, and that's great because the primary constituent of a lot of these extracellular fluids is water. We will also see those hydrophilic heads facing inside of the cell, which is also good because the cytoplasm or the jelly of the cell is primarily water as well. We will also see some glycolipids, and the remainder of that boundary will be cholesterol. Cholesterol is important in keeping that membrane stiff and making sure it actually has structure to it. Scattered along this phospholipid bilayer, we will see different types of proteins. Later on, these proteins we will see are the doors and windows that will allow substances to either exit or to enter the cell. Pictured on this slide, we are looking at an up-close view of the plasma membrane of a cell. You are able to see the phospholipid bilayer. You have an outer layer of those phospholipids as well as an inner layer. And if you recall that the head of the jellyfish or the main candy part of the lollipop faces outside as well as inside, and that's because the primary constituents of extracellular fluid and the cytoplasm is water. So those heads, or those hydrophilic heads of a phospholipid, are water loving. So they are attracted to those fluids. You are also able to see membrane proteins. So those are going to be the larger purple structures that are placed along the plasma membrane. We will see those proteins serve as doors to allow substances to move into as well as out of the cell. Still talking about the plasma membrane structure, as previously said, those proteins allow for the cell to communicate with the surrounding environment. But furthermore, we have two different types of proteins. If you revisit the previous slide, you are able to identify integral proteins as well as peripheral proteins. Those integral proteins you could think are firmly integrated or inserted into the membrane, and they are oftentimes transmembrane proteins, meaning they span the entire width of that phospholipid bilayer. Those integral proteins will serve as transport proteins, also known as channels or carriers. Those are the ones that we will see serve as gates or doors to allow for the cell to communicate with the external environment. The other type of protein is known as a peripheral protein, and as its name suggests, will be located along the periphery. So usually peripheral proteins are not transmembrane proteins. They are simply found hanging off of the outside or attached to the inside. Now, these plasma membranes don't just let anything enter the cell. They don't always let something leave the cell. So they are considered to be selectively permeable, meaning they can pick and choose what they allow to permeate through that wall. If something is highly permeable, it will let things pass through with ease. Whereas if something is impermeable, substances and molecules are unable to move through that plasma membrane. Two different ways that we will see substances pass through the plasma membrane. We have passive and active. And again, as their name suggests, passive processes will not require energy. They will just simply occur. Active processes will require energy in the form of ATP. We will first start by talking about passive processes or passive membrane transport. And again, passive processes do not require ATP or energy. And we can identify two different types of passive transport, the first being diffusion. We have several different types of diffusion, but in short, diffusion will occur when a substance moves from an area of high concentration to an area of low concentration. And the other type of passive transport is known as filtration. We will not talk about this in this chapter, but it usually is seen occurring in capillaries, and capillaries are very tiny and small blood vessels. Substances will passively diffuse, meaning it doesn't require energy, down its concentration gradient. And this gradient is the difference between the area of high concentration and the area of low concentration. Substances will readily diffuse from an area of high concentration to an area of less concentration. And again, that does not require ATP. The speed at which this process occurs is influenced by several factors, such as the molecular size. Usually, smaller molecules will diffuse more rapidly, and temperature is also another factor. As you increase the temperature, or a higher temperature will allow for, again, a more rapid rate of diffusion. The example seen on this slide might remind you of maybe, again, simple diffusion occurs without energy. Molecules or substances will naturally diffuse down their concentration gradients from an area of high concentration to an area of low concentration. And we will see certain substances like sodium and potassium pass through that plasma membrane down their concentration gradients, not requiring energy. But because of those membrane proteins and those gates and those channels, the plasma membrane does stop diffusion and is responsible for creating these gradients. If this was not the case, or if this membrane was not in place, these substances would always be in areas of low concentration. Another type of diffusion or passive process is known as facilitated diffusion. There are certain molecules throughout the body that are rather large, as well as hydrophobic, or afraid of water, basically. Glucose is a great example, but the molecules listed here are going to use either a special carrier or a channel. And keep in mind that this is still diffusion. These facilitators, like carriers and channels, are still going to allow molecules and substances to diffuse down their concentration gradient. A carrier-mediated facilitated diffusion is a membrane protein that will grab onto one of these molecules and alter its shape, ultimately allowing for this molecule or this substance to pass through the plasma membrane down its concentration gradient. A channel-mediated facilitated diffusion is a water channel. So certain lipid-insoluble solutes or other water molecules are allowed to move through these channels. Moving away from passive processes and beginning to discuss active processes or active membrane transport, recall that these processes require energy in the form of ATP. Two different types of active processes. We have active transport as well as vesicular transport. Either way, when substances use active membrane transport, it's usually for any of the following reasons. That solute may not fit through a channel, it may not be lipid soluble, or that solute might not be able to move down its concentration gradient, meaning it cannot move from an area of high concentration to an area of low concentration. These solutes will require carrier proteins, or basically something to hold its hand so it's allowed to cross that plasma membrane. So some of these carriers are capable of transporting more than one substance or more than one molecule at a time, which is extremely efficient. And we can name the carrier based on the direction that it is transporting those substances. An antiporter will transport different substances in opposite directions. So you could think of this as you and someone else are walking through a door heading in the opposite direction, and you simply wave to each other. The other type of carrier protein is known as a symporter, meaning those substances are moving in the same direction. Either way, this active membrane transport moves solutes against their concentration gradient. It's pushing them in a direction that they don't necessarily want to be in. Therefore, this does require energy. It's like an uphill battle. And we need those substances to move against their concentration gradient, not because that's where they want to go, but because that's where they need to go to maintain homeostasis. We also have two different types of active membrane transport, primary and secondary. For the most part, we will only address primary active transport, and we refer to it as primary because the energy that is required to transport those substances comes directly from that protein or from that membrane protein. It doesn't receive it from elsewhere. It's able to create it right there. So again, primary active transport, we will see these channels are going to phosphorylate or change shape to allow for substances to be pumped across the membrane against their concentration gradient. And the most popular example of an antiporter, two, primary active transport, is known as the sodium-potassium pump. For the next few slides, we will focus on the sodium-potassium pump. Again, it is an example of an antiporter, meaning that it transports different substances in opposite directions. And it is also the best or most studied example of primary active transport. It's primary because ATP, or energy needed to function that pump, comes directly from the pump. It's called the sodium-potassium pump because it's responsible for transporting sodium and potassium. The sodium-potassium pump is going to transport sodium out of the cell and potassium into the cell. Remember, those substances are moving against their concentration gradient. So as it pumps sodium out of the cell, it's pushing sodium into an area of higher concentration. And the same goes for potassium. As that pump forces potassium back into the cell, it's moving it into an area of high concentration. We will revisit this cell when we talk about the muscular system, and you will also see it in EXS217 when you talk about the nervous system. But both of those systems involve excitable cells. So we will see the sodium-potassium pump in those systems, but you should note that the sodium-potassium pump is essentially found in all plasma membranes of all cells throughout the body. You should note that also along the plasma membrane we have what are called leakage channels. And as their name suggests, they leak. They are constantly open. They allow for substances to flow down their concentration gradient, and they do allow for sodium and potassium to leak into the direction that they actually want to go. So we will see sodium leak into the cell, and we will see potassium leak out of the cell. This pump is in place to put those two substances where we need them to maintain homeostasis. And lastly, the sodium-potassium pump helps to maintain this electrochemical gradient of sodium and potassium. The electrical gradient refers to the voltage. So overall, the inside of the cell is generally more negative compared to the outside. And you could also look at the chemical concentration when you look at the electrochemical gradient. Again, that just refers to the concentration of substances. On this slide, you will see the steps or the processes that are involved with the sodium-potassium pump as it transports sodium and potassium against its concentration gradients. At the very top, you see 3 sodium binding to the pump. Those are the little yellow balls. 3 sodium bind to the pump. That ATP, or that energy molecule, hydrolyzes and eventually opens the pump up to the extracellular fluid. The pump then kicks those 3 sodium off to the outside of the cell, and as it's open to the outside of the cell, it brings in 2 potassium molecules. Again, the pump phosphorylates or changes shape and reopens to the inside of the cell to release those 2 potassium molecules. And as it does that, it resets itself. So we have another energy molecule attach itself, and it is ready to grab 3 sodium molecules. So overall, you need to know how many sodiums and how many potassiums are being transported with this pump. It's not 1 and 1, it's actually 3 sodium molecules and 2 potassium molecules. In addition to that, you need to know the direction that the pump moves them. We have 3 sodium molecules moving out of the cell and 2 potassium molecules moving into the cell. The other type of active transport or primary active transport is known as vesicular transport. We will use this type of transportation for larger particles as well as macromolecules, and we will also see fluids travel into and out of the cell using vesicular transport. We get the term vesicular from these membranous sacs that we package these particles in, known as vesicles. You could think of them as little bubbles. Either way, this type of transport also requires energy in the form of ATP. We will see several different types of processes utilize these vesicles. We have endocytosis, exocytosis, transcytosis, and vesicular trafficking. The 2 that we will focus on for the most part, the first 2, endocytosis, we are transporting something into the cell, hence the term endo. Exocytosis, we will move stuff out of the cell, hence the term exo. Endocytosis is, again, the transport of something into the cell. Endo means inner, cyte or cyto means cell. On this slide, you see the different steps that are necessary for endocytosis. However, I just want you to know a very general idea that endocytosis is the transport of something into the cell. The top left of the diagram, you see that we have a coated pit. So this pit is really kind of formed by the plasma membrane. It ingests the substance and it's going to carry it throughout the cell. From there, the cell can do several different things with it. If you look at step 6, we can either destroy the substance using an organelle inside of the cell, or we can actually transcytose it, meaning we are taking that substance into the cell and simply passing it through the cell to get rid of it on the other side. There are 2 types of endocytosis. They're going to differ in the type as well as the amount of material taken up and really the means of uptake. The first type is known as phagocytosis, and we think of this as cell eating. We will see phagocytosis when a cell is going to ingest a large or solid material like bacteria or cell debris. Phagocytosis is typically used by cells that are going to destroy whatever it has ingested, like white blood cells. Pinocytosis is known as cell drinking. So pinocytosis, we will see a cell bringing in extracellular fluid as well as some dissolved solute. And a cell will use this to sample the surrounding environment. And oftentimes, the cells of the small intestine will utilize pinocytosis because the small intestine's primary function is nutrient absorption. On this slide, you will see 2 diagrams of the different types of endocytosis. Either way, they are both ingesting something and bringing it into the cell. On the left, you have phagocytosis, which we will use for larger material such as bacteria or debris. And on the right, we see pinocytosis, often referred to as cell drinking. The cell will ingest some of the fluid and dissolve solute into the cell to sample the environment. Another type of vesicular transport is known as exocytosis. This type of transport occurs when we kick something out of the cell or eject it from the cell. So this is really the secretion of a substance from a cell. That secretion can be hormones, neurotransmitters, as well as mucus and cellular wastes. Prior to these substances leaving the cell, they are enclosed in a secretory vesicle. Secretory meaning secretion. This secretory vesicle within the cell will fuse to the plasma membrane or connect to it, and it will release its contents to the exterior. On this slide, you see a schematic or the steps that are necessary for exocytosis. You do not have to go in great detail with all of the big words on this slide. However, you should understand that exocytosis is the transport of substances out of the cell. Usually, these secretory vesicles will contain a secretion like hormones or neurotransmitters, which are simply chemical messengers. That secretory vesicle will move about inside of the cell until it fuses or binds to the plasma membrane. It will open and it will release its contents to the exterior. The last thing we need to address with regards to the plasma membrane of a generalized cell is this membrane potential. This membrane potential exists at the plasma membrane and is due to or is produced by the separation of these charged particles. Remember, sodium and potassium both carry with it a positive charge. When they are in different areas, it establishes this membrane potential. When a cell is resting, meaning it is not excited, like a nerve cell or a muscle cell, it is said to be polarized. Because of this sodium-potassium pump, along with other factors, we have this difference in charged particles. Overall, the interior of the cell is generally more negative compared to the exterior of the cell. The main factor in establishing this resting membrane potential, or the membrane potential, is due to potassium. So if we take into account the sodium-potassium pump as well as these leakage channels, we will see potassium leave the cell through the leakage channels down its concentration gradient, which is awfully steep. So as potassium leaves the cell, it takes with it its positive charge, making the inside of the cell more negative. Now, again, opposite charges attract. So as the interior of that cell becomes more negative, when potassium diffuses down its concentration gradient, the negative interior then pulls that potassium back in. And as we have that back and forth pull, it is truly balanced, and we could say that the cell is resting, or we have a polarized cell, or the membrane potential. We are now going to shift gears and move away from the plasma membrane of cells and begin to talk about the second component of a generalized cell, known as the cytoplasm. If you recall, the cytoplasm truly includes everything between the plasma membrane and the nucleus, or the command center of the cell. This is where the majority of cellular material is, and it is also the site of most cellular activities. But the stuff in between the plasma membrane and the nucleus includes the cytosol, which is a great example of a colloid or a gel-like solution. We have some inclusions in there, such as glycogen, color pigments, or lipids, depending on what type of cell you are actually in. And lastly, organelles. These are the little organs that allow cells to function and to carry out life. We will primarily focus on the organelles when it comes to the cytoplasm, or the little organs. And we will see that some of these organelles have a membrane or an outer boundary that allows for compartmentalization. We have the mitochondria, the endoplasmic reticulum, the Golgi apparatus, and we also have non-membranous organelles, or organelles without a boundary or membrane. And we will address ribosomes when we get there. As far as any of these organelles go, you should really know their function and not so much their structure, maybe a little bit, but a simple matching question could take care of this on an exam, so make sure that you are familiar with the definitions or the functions of each organelle. The first one, the mitochondria, we think of as the powerhouse of the cell, and in the simplest of terms, it's going to produce energy, also known as ATP. And it does so via aerobic cellular respiration. Aerobic meaning it requires oxygen. Next, we have the endoplasmic reticulum, which will appear like a stack of dinner plates or parallel interconnected membranous tubes. We have two types of endoplasmic reticulum, and we name them truly based on their appearance. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is deemed rough based on its appearance. It has this studded appearance because it contains ribosomes. Slightly further down, a few bullet points, you will see that we will talk about ribosomes, but for now, the rough endoplasmic reticulum is where we see the synthesis of proteins. These proteins produced by the rough endoplasmic reticulum will be used in the plasma membrane. Those proteins will contribute to that phospholipid bilayer. Ribosomes, if you remember from earlier, were a type of non-membranous organelle. But the reason that the rough ER is responsible for protein synthesis is because of the ribosomes. Ribosomes are a site of protein synthesis. Smooth ER, or smooth endoplasmic reticulum, appears to be smooth because it does not contain ribosomes. We will see smooth ER responsible for the metabolism of lipids or fats, as well as cholesterol. We will see the smooth ER detoxify certain chemicals, such as drugs or alcohol. And we will also see smooth ER convert glycogen to glucose, as well as store and release calcium. So again, these definitions are extremely important for your exam. On this slide, you are able to see both smooth as well as rough endoplasmic reticulum. If you look at the blue structure, so the flattened membranous tubes or sacs, you see the rough ER containing ribosomes, or those red or pink studded structures. Again, the rough ER containing the ribosomes is the site of protein synthesis. Towards the top of the screen, you see the smooth endoplasmic reticulum. It does not contain ribosomes. We will see the smooth ER play a role in lipid metabolism, cholesterol metabolism, transporting glycogen into glucose, as well as the storage of calcium. So be sure that you know the definitions, or really the difference between smooth versus rough ER. The Golgi apparatus. This may look like a bit of information on the slide, but again, simply just know the definition. I like to think of the Golgi apparatus as the Postal Service. The Golgi apparatus will receive proteins and lipids from the rough endoplasmic reticulum, so it receives the package. From there, it's going to modify, concentrate it, and then package it up to send it elsewhere. And once it does that, once it finishes modifying, concentrating, and packaging, it could send it 3 different ways. You do not have to know these 3 ways in detail. I would just understand the Golgi apparatus as the postal service. Again, you won't see that on your exam. So it modifies, concentrates, and packages substances received from the rough ER. On this slide, the Golgi apparatus is pictured here in green. You can see that it will receive a vesicle or a bubble from the rough endoplasmic reticulum in blue. From there, it takes the substances, whether it's the lipids, proteins, cholesterols, transports it through the tubes, modifies it, concentrates it, and then we'll send it elsewhere. This slide shows you the 3 different pathways that the Golgi apparatus could send a vesicle once it's done modifying it and concentrating it. But again, you do not have to know the 3 pathways, simply understand the definition or the function of the Golgi apparatus. We will switch gears briefly and talk about cellular extensions. However, you should note that these are not organelles. They are not necessarily associated with the cytoplasm, but there are structures in the cytoplasm that will anchor these extensions. Certain cells throughout the body will have things coming off of them or structures coming off of them. The first couple are cilia and flagella. Cilia are responsible for moving materials across the surface of the cell. You could think of cilia as little hair-like extensions. Usually cilia will move mucus, especially in the respiratory system. Flagella, however, will aid in the physical movement of the entire cell. We will see flagella with a sperm cell, so it's a whip-like tail that is responsible for moving the sperm cell throughout the body. Another cellular extension is known as a microvilli. Microvilli are finger-like projections. They do not move the cell. They do not move substances across the surface of the cell. They are simply there to increase the surface area. You will see microvilli in the digestive system. To increase the surface area to ensure nutrient absorption. And the last component of a generalized cell is known as the nucleus. We will talk a little bit about its function on the next slide. We will study a picture of it, and then we will be all done. So again, the nucleus is the last component of a generalized cell. And things don't really work without a command center or something controlling them, and that is what the nucleus provides for cells. It contains DNA, and it also provides commands for the entire cell to function. Usually cells will only have one nucleus. We can consider a cell with one nucleus to be uninucleated. But sometimes we will see cells that have many nuclei, and we would call it multinucleated. Examples of multinucleated cells or cells that require greater control would be skeletal muscle cells, for example, the cells that make up your biceps brachii muscle. And we will also talk about cells that have no nucleus or are considered anucleated. The best example is a red blood cell found throughout the cardiovascular system. And we will finish up by looking at this picture of a nucleus, and it's really that large purple or magenta ball. You can also see the rough and smooth ER just outside of it, but that nucleus is really the cell center where it will provide all of the commands to allow the cell to function appropriately. If at any point in time you have questions throughout this lecture or after you're done watching it as you are addressing the objectives and questions, please do not hesitate to email me. It is johnsonml@uncw.edu.